Class will see all about how the self-assembled nanostructured materials are used for various applications. In fact, we have seen in one of the concept called like protein structure, like we have seen secondary structure, tertiary, and quaternary. So when the protein is getting interacted with the aqueous media or some other medium, they start to, I mean, assemble into certain structure, maybe of alpha helix or beta sheet or some sort of sub nano unit, all those things. So those structures, how it can be taken it for further, I mean, useful biomedical applications. We'll see. So one of the, I mean, kind of general explanation is that self-assemble is the spontaneous organization of smaller subunits to form either large or well-organized patterns. Okay? Say, for example, you have surfactant here, like they have hydrophilic head and hydrophobic tail portion. And the moment when you start to mix them with the aqueous media, okay, you know that like sugar and water. Like you take a 10 gram of sugar and if you are dissolving them in water, it dissolves. Then meanwhile, you take 5 kg of sugar and you dissolve them in 1 liter of water, they will not dissolve. Of course, if we start to heat, I mean, they'll start to dissolve. So what will happen as a result if you're dumping it all of a sudden, then there'll be a formation of small lump section. You can find it. So that same thing used to happen. Look, surfactant, the moment when you start to dissolve them in the water, they will dissolve of course. But yeah, the more when you want to dissolve more amount of surfactant in the same aqueous media, they will not dissolve. Rather dissolving, they'll start to form small aggregates or the lumps. Okay, and those aggregates are usually called as a self-assembled structures. Okay, so likewise, I mean, you can see here one of the structure like you have one hollow architecture. On top of that, those I mean, a number of number of surfactants are eventually they I mean formed such a way that this kind of well organized structure has been created or the formed. Okay. Now this hollow space, since you have this kind of well defined spaces, you can use them for either kind of drug encapsulation or other kind of something like gas separation purpose, etc. You can use them. And the other concept, like once you have this kind of nanoparticles, again you can use make them use for self assembly of those nanoparticles with the help of proper functionalization. Okay, so here you can see one of the example called like you have on gold nanoparticles. On top of that, I mean, they have functionalized those nanoparticles with the help of different functionalities like oligonucleotide or thiolination, etc. Okay? And basically, those design is used to target certain RNA sequences. Okay, so once if your functionalization is, I mean, depending upon those targeted RNA, what will happen? The moment when you keep this self-assembled structure nanostructure into that media or the sample where you want to detect, so they'll start to detect those RNA, and once the RNA is matches with those your surface functionalities of your nanoparticles, what will happen? They'll start to release those frozen ions. Okay, and once the frozen ion is started to release, what will happen? With the help of microscopic structure, you can detect what is coming out. If the frozen signal is coming from your sample, that means your sample, I mean, has pathogen or sample, whatever, has so and so, I mean, RNA sequences. Okay, and depending upon the intensity of the signal, I mean, you can you'll get to know like how intensity of your, disease is there in your sample or the body. Okay, so yeah. So when the targeted RNA, I mean, binds to the nucleotide of the surface of the self-assembled structure, the fluorophore or the fluorescent ions start to release from the structure, and it becomes clearly visible in the fluorescent microscope. Okay, and this is one of the same example they have used to for detecting cancer cells. Okay, so what they did, like gold nanoparticle, again they have decorated, functionalized with the help of same RNA sequences of the cancer cells. Okay, once the cancer cell, I mean the sample, once they introduce the samples into the self-assembled one nanoparticles, what will happen? The RNA, I mean, from the cancer cell line, they will start to absorb on the surface of self-assembled gold nanoparticles. As a result, your, I mean, gold nanoparticle, they release fluorescent ion, okay, from the oligonucleotide, and yeah, subsequently that can be detected easily from the fluorescent microscope or any other kind of laser microscope. And if there is no those RNA sequences, if they are not matching, then there will be no fluorescent ion release, and of course we are not able to see any kind of fluorescent, I mean, signal from the microscopic image. Okay? And here the left side images shows the fluorescent signal. That means comes from the cancer cell, and the right side shows the healthy cell, means like normal cell line. That means there was no, I mean, RNA sequences which are matching with the functionalized gold nanoparticles. And you can also use for other kind of MRI scanning as well. Like you can use like kind of magnetic nanoparticle. People usually use them. So instead of using magnetic nanoparticles, you can also use some other, I mean, self-assembled fluorescent blocks as well. Like because, I mean, of course, if you use both like together, I mean, take a magnetic nanoparticles and then you functionalize them with the help of different oligonucleotides and all, then again your intensity of detection will be better actually. I mean, the contrast will be pretty much high. Even the small amount of either cancer tumor or the cancer cell, if it is affected to the specific tissues, can be detected very easily. So again, this kind of once you have this kind of self-assembled structure, you can, enhance the drug loading efficiency of the carrier. Okay, because always loading efficiency is preferred. I mean, higher loading efficiency is preferred because it can be used for longer time. When provided, yeah, if you could be able to release them in the controlled manner. Okay. So how you can release, like, since you have more number of spaces, I mean, in the architecture of your, either miser layer or kind of those, hollow architecture. Okay. So as a result, like more amount of ducts can encapsulate into the spaces between this miser layer or the kind of coastal particles. Well, this is how self-assembled nanoparticle usually, I mean, does. I mean, drug release characteristics, like this was the standard diffusion based on the, normal carrier, and this was nano-assembled, like a self-assembled nanoparticles. Encapsulated. Okay, I mean, you will have those kind of controlled drug delivery. I mean, either it will not, I mean, burst or it will not terminate it so slowly. Okay. Also, you can target some of the tissues as well. Okay, say suppose if I want to, use some of the drugs for, I mean, killing the cancer cell, what will happen? Like, that how cancer cell DNA sequences or the RNA sequences of the cells. Okay? So what we can do, you make such a way that your drug encapsulate carrier, you make such a way the functionalities like same like of RNA or the DNA, sequences of the cancer cell line. You plan accordingly so that they start to bind, directly to the cancer tumor, and subsequently, of course, once they enter the cancer tumor cell line, there will be a different environment, like there will be a changes in the temperature and the pH, etc. So that will disturb the drug and the carrier interactions. Of course, once that is disturbed, then the drug will start to release slowly. Okay. So that is called targeted drug delivery to the specific tumors. And yeah, this is again like alternative to nanoparticles, okay, or the surfactant, which is called self-assembled structure. Right, like how the micellar usually works, like same like due to their low solubility, because with the moment when you take surfactant with a higher amount of quantity wise, they are not dissolved in the aqueous media or the water media, where you want to dissolve your surfactant. So at the highest concentration, wherein all the molecules are in the dispersed state, or it forms as a kind of small aggregate. Okay. So the concentration beyond which, why only micellar formation is possible. Okay. Below that, of course, your surfactant can be, I mean, completely soluble. So beyond which the surfactant will not be able to dissolve, and as a result, there will be a formation of small aggregate, or sometimes there should be in the form of dispersed state. That is called, at that point is called a critical measurable concentration. Of course, this, is like they have a long, I mean, aliphatic chain. I mean, that is called tail, and you have a, I mean, polar region which is called like head of the micelle. So yeah, so the picture here, they dissolved well, and then slowly they started to form as aggregate. And itself, if you keep and add more amount of surfactant, you will end up with this kind of self-assembled micellar structure. So, so yeah, so this micelle can be targeted. I mean, targeted in the sense the size of the micelle can be prepared in the size even 10 nanometer in size. So that means the diameter of the micelle. So as a result, even the hydrophobic drugs can also be encapsulated in the core section of your, I mean, micelle, and that can be targeted. I mean, delivery can be used. And this is some of the microscopic images of the micelle. Like you can see here, I mean, formation of this shell, and you have a core. Okay, this suppose this is, I mean, pure micellar, the control micellar. So if you want to encapsulate some of the drugs into the structure, okay, what you can do, you do load the drugs. Again, you prepare the sample for the high resolution translucent microscope. Then you'll get to know how much the course was, I mean, which was closed in the microscope. And yeah, you release the drug again, you can, I mean, collect those micellar sample, and then you can check for the microscopic image, and then you get to quantitatively how much the drug was encapsulated and how much it was released. So so far fine, or you have any issues with the concept? It's okay, right?
Will you please repeat the part where the targeted drug delivery of tumor was explained?
Okay, all right. So what is hydrogel? Hydrogel we have seen already, right?
Yes, sir. It was explained for the drug delivery system, how it's
What
used.
is hydrogel? Hello. Well, hydrogels are nothing but kind of gel-like structure, three-dimensional structure, where you have 90% of water and 10% of your materials called either polymer or the protein. So you need to cross-link those hydrogels so that you can use them for, I mean, for any kind of shape fertility purpose in tissue regeneration. Sorry? Hi, who has asked?
So,
Yeah, yeah. Go ahead. What is your question?
how the part where
I'm not hearing your voice.
targeted tumor part, tumor. So let me
Hello.
type.
Maybe I have some little question. Hello. Hello.
Yes, sir.
Yeah. You were asking some doubt, right? I
Yes, so the part where tumor was explained, I didn't get it clearly. So can you elaborate on it a little bit?
just wait.
So
Wait. I'll
how
share.
the procedure takes place?
Just wait a minute. Yeah, here, right?
Yes, sir.
This So what we can do, I mean, what people usually does, like they prepare the self-assembled structure and then they load the drugs. So before that, they knows that I mean they knows the DNA or RNA sequences of the cells, right? I mean the amino acid sequences. So they'll design such a way that I mean the same to that of your nanoparticle structure. I mean surface functionality. So once they do that, what will happen? I mean the whatever the drugs the patient they are taking oral way, they'll directly start to get attracted to the tumor. I mean, because others, I mean, tissue or the organ or the other cell lines, they will not have the same amino acid sequences. Okay, as a result, these people, I mean, the drugs encapsulated self-assembled nanostructure, they'll directly attracted towards to that tumor. So once the carrier encapsulated drug, of course, the altogether once they get into the tumor, they'll experience a different environment. Of course, cancer tumor has, I mean, above like 40 degrees Celsius, okay, surrounded tissues as well. And they that is enough to, I mean, break those bond. I mean, if it is physically loaded drug, I mean, of course, hydrogen bond or any other secondary bonding. I mean, that temperature or the pH can break those bond and subsequently the drugs will start to release on the tumor surrounded area. So, are you getting it? So,
Yes, sir. That's clear. Also, is there any involvement of any virus or something in this procedure, in this process?
involvement of
Uh, Virus, like in the case of this other example, there was involvement of virus. Harmless virus is used for to target. So, is there the same thing here as well?
yeah, you can use that route as well. I mean the common the concept is like that. You have to plan such a way that your either oligonucleotide or RNA sequences has to match actually. So that they can directly reach those concerned organ or the tumor or the tissue, whatever. So in your doubt, like then you have to plan such a way that the virus where which I mean the type of virus can directly reach to that concerned tumor. So that kind of screening you have to do it. Are you getting it?
Yes, sir, clear.
So hydrogels, you have seen it already. So I just giving you the some of the glimpse of the applications. Like it can be used for cartilage tissue regeneration as well as for I mean kind of other tissues like I mean muscle skeleton tissue regeneration as well as for even hard bone tissue regeneration. Okay, and yeah, since it is a very soft material, with the help of different cross-linking route, like if you can cross-link more with the help of like either photo cross-linking or kind of, chemical cross-linking. You can enhance the crossing density, and subsequently they'll enhance the mechanical stability of the hydrogel. So once you do that, then it can be used for any kind of, tissue regeneration, not only for cartilage. Fine. So we have seen iron nanoparticle, which has excellent magnetic properties. Okay, and we have been used for different sort of applications in biomedical sector, like magnetic imaging, the magnetic heating called hyperthermia, and of course targeted drug delivery and detection and purification and isolation of proteins and their manipulations, etc. So sorting in the sense you're gonna, separate certain types of cells, okay, from the mixture of, cells, okay. So aim is to detect or separate or isolate one type of cell from the others, and often when the target is present in very small quantities. So assume that you have a RB flask or any kind of, cell culture dish, and you have a different cells and you have a different, of course, targets. Okay, so what you're going to do? So you're gonna uh, decide the ligands, which directly, get connected to that concerned cells. Okay, so that ligand you got to select, and concerned ligand you're gonna, functionalize on the surface of the magnetic nanoparticle. So once you do that, okay, so once the functionalized, magnetic nanoparticle, okay, they'll start to attract to that specific, cells, which has, specific target, okay, from the other cell lines. Then of course you have a magnetic, attached magnetic particle attached to cell lines. So then with the help of magnetic, forces, you can straight away, isolate those cells from the mixture of cell lines. Okay, and the another concept is called hyperthermia. Okay, so hyperthermia is one of the method where you can, use to kill the cancer cells or the cancer tumor in the beginning or the stage one, phases. Okay, so what will happen the moment when you apply the external magnetic field, to that magnetic nanoparticle? What will happen? They'll enhance the temperature surrounded area. Okay, maybe of like a 42 to 46 degrees Celsius. Okay, and at those temperature, like 42 to 46 degrees Celsius, the cancer cell growth is either stopped or they get slowed down. Okay, so what we can use, what we can do, you take a magnetic nanoparticles, okay, and then you functionalize them, with the help of any kind of antibody or the kind of ligands, such a way that they directly, attract towards the cancer tumor. Okay, this all will be, absorbed through the microscopy or the MRI scan. So once the magnetic particles, either decorated by the drugs or the ligand, which once it is attracted towards to the cancer tumor, okay, then you start to apply the magnetic field, surrounded to that of tumor. Okay? Then once the magnetic particle experience the field, the temperature will start to rise. That is called hyperthermia, or it is called overheating. So once the tumor reached 42 to 46 degrees Celsius, what will happen? The cancer cell will start to die, or the growth will be stopped. Okay, and yeah, can be used to, kind of either tablet or oral, way, etc. And yeah, like embedding means like they used to embed it in the biodegradable polymer, and then they used to take it to that of tumor where the cancer cell ends are present. So that way also you can do. And yeah, this is how, like you have kind of cancer tumor, and then the drug was given, by injected way. Once the drug is injected, which has the magnetic nanoparticles also, so you can take them. Of course, the nanoparticles modified surface has a tendency to get attracted towards the the cancer tumors. Even if they don't attract it with the help of magnetic field or the magnetic, playing magnets, we can align those drugs embedded magnetic nanoparticles into the cancer tumors. and then you apply a slightly higher magnetic field, then they'll, enhance the heat will start to enhance or the temperature, and that is enough to stop the cancer cell growth or the complete, dissociation of the cancer tumors presence in the human body or the animals. Okay. Well, this can be used in the stage one because, of course, we do have various anti-cancer drugs which has different kind of toxicities because they harms other tissues as well as organ of your body. Okay, and this is, targeted only, to the specific cancer tumors portion. So as a result, like we can avoid the affecting of other tissues or the organs in the body. Well, we do have some of the other, issues or the limitations, kind of like dispersion issues and the biocompatibility of the magnetic nanoparticle. All those things are there. So that's the reason, like, with stage one, it is very easy to use because there'll be a very small tumor where you can, completely you can kill away. If it goes stage two, again we'll have to use plenty of drugs, etc. As a result, like you will have to use large amount of magnetic nanoparticle, and since they are not completely biocompatible, for the long time, like that this can be, does some issues with the health of the patients. The rest other stuff like some other types of drug loading as well as releasing system is there in the iron oxide based nanoparticles. We'll see in the next class. And then you have to write one more quiz also, right? When you want to write next week Friday? You have given 2 assignment, right? So far.
Yes, sir.
Yes, sir.
Okay, write one more quiz, then I'll check. Quiz one was, too bad. I'll send you the marks by this week, and if everyone is feeling, then we will have to write one more quiz. Like out of 3 quiz, I'll take the best 2. So
Okay, sir.
this Friday you are available. Sorry, the next Friday is available. You have the end semester exam, right? Monday also you can write, although we don't have class. So now we are here, right? Yeah. Oh no, you have Diwali also next Thursday. Okay. So this Tuesday, can you write? Number 2.
During class timing, sir.
No, we can have it other